All right, welcome to the Explainer. Today we're diving into a really fascinating bug, Pediococcus acidilactic, a bacterium with a serious identity crisis. Is it a good guy or a bad guy? Well, that's what we're here to figure out. Now, everything we're about to cover was pulled together using a Gemini deep search. And for all your fellow microbiologists out there who love the nitty-gritty details, don't worry. All the sources are linked in the notebook document that goes with this. So let's just get right to the heart of it. Probiotic or pathogen? Is this the helpful bug you find in supplements and fermented foods? Or is it something that should have us worried when it pops up in the lab? Well, the short answer, and what makes this so interesting, is it's both. Okay, so here's how we're going to break this down. First, we'll talk about its double life. Then we'll get into how to spot it in the lab. After that, we'll look at when it's a friend and when it's a foe. Then comes the tricky part, the treatment conundrum, and we'll wrap up with how to manage and contain it. All right, Section 1, a bacterium's double life. Let's start with an introduction to this pretty unique gram-positive coccus. See, Pediococcus acidilacti is part of the Lactobacilliaceae family, and it has this total split personality. On one hand, it's a super common probiotic used in the food industry, but on the other, it can be a really serious opportunistic pathogen. Next up, unmasking the organism. Let's talk about the key characteristics for nailing the ID in the lab. So, how do you actually spot this thing? Well, there are a few key clues that should immediately set off alarm bells, telling you, hey, this isn't your run-of-the-mill Enterococcus or Staphylococcus. Okay, first things first, let's look under the scope. The biggest giveaway is its morphology. You're looking at gram-positive cocci, but the real tell is how they're arranged. Because Pediococcus acidilacticus divides on two different planes, you get these absolutely classic, textbook-perfect tetrads. I mean, they are just beautiful and so distinctive. Now, what about on the plate? It's a facultative anaerobe, grows just fine on your standard sheep blood agar, nothing too wild there. You'll see these small, white, opaque colonies. But, and this is the really cool part, its key superpower is that it's thermotolerant. The fact that it can grow at 50 degrees Celsius, that is the critical test that separates it from its very close cousin, Pediococcus pentosaceus. And this table right here, this is where we create the biochemical fingerprint. It's so cool how a few simple tests can just lock in the ID. So think about it. You've got a gram-positive coccus that's PIR negative, inherently vancomycin resistant, and grows at 50 degrees but doesn't ferment maltose. Boom! That is a massive clue pointing you straight to Pediococcus acidilactici. And right away, that whole pattern tells you it's definitely not an Enterococcus. But sometimes the classic methods just aren't enough. When things get a little fuzzy, we bring in the big guns, molecular. Sure, 16S rRNA sequencing works, it's reliable. But if you need a fast answer to tell it apart from Pediococcus pentosaceus, a multiplex PCR for the LDHD gene is the way to go. It's super specific. And for those really critical cases, like the endocarditis we're going to talk about later, the absolute gold standard is sequencing the 16S, 23S intergenic spacer region. You just can't beat that level of resolution. Okay, part 3, friend or foe? Let's get into the clinical significance of Pediococcus acidilactici. We know how to find it, but now for the big question. What does it do to people? And trust me, this is where the plot really thickens. I love this slide because it just lays it all out. The two faces of this bug. On one side, the friend side, it's producing these things called pediocins, which are antimicrobial, and it's even helping to modulate our immune system. Pretty cool, right? But then there's the foe side, causing bacteremia, sepsis, even endocarditis. And here's the real kicker. Its greatest strength as a probiotic, its toughness, its ability to survive just about anything, is the exact same trait that makes it such a dangerous pathogen when it gets the chance. So what do these infections actually look like? Well, the most common thing we see is bacteremia or sepsis. And a lot of the time, it's not alone. It's part of a polymicrobial infection. But honestly, the thing that really gets our attention, the thing that makes you sit up and take notice, is its potential to cause really nasty, deep-seated infections like endocarditis, especially in patients who have prosthetic heart valves. And that brings us to our next section, the treatment conundrum. This is all about its intrinsic resistance and, more importantly, the total absence of guidelines. So, what do you do? How do you treat an infection when the rulebook is basically empty? Okay, this is absolutely fundamental. We have to be crystal clear. This is not acquired resistance. This bug doesn't just learn to fight off vancomycin. No. Pediococcus acidilacticus is born this way. It is intrinsically, fundamentally resistant. Why? Because its cell wall ends in D-alidilactate, not D-alidila. Vancomycin literally has nothing to grab onto. The drug is completely useless against it from the get-go. And this right here is probably the single most important slide for anyone working in a clinical lab. A direct quote from UCAST. There are no species-specific clinical MIC or disc diffusion breakpoints for Pediococcus species. Just let that sink in. What does that mean for us? It means when we run a susceptibility test, interpreting the results is a huge challenge. There's no magic number that tells us if it's susceptible or resistant. So with no breakpoints, what's the protocol? Well, UCAST is very clear on this. Step 1, you report only the quantitative MIC. No interpretation, no S, I, or R, just the number. Step 2, you have to tack on a standardized comment that basically says, hey, we don't have breakpoints, so we don't know what this number means clinically. And step 3, and this is the most important part, you push for a clinical consultation. The lab's job isn't to give an answer, it's to start a conversation. Okay, section 5, management and containment. So with all these challenges, how do we actually handle this thing? Well, we have to build our own playbook using case reports and what we know from VRE protocols. So when it comes to treatment, we're basically flying by the seat of our pants, guided by case reports. If you're isolated susceptible, penicillin or ampicillin seems to be a go-to choice. For something really serious like endocarditis, there's a case where they used daptomycin for a full 42 days, and it worked. Now, for my friends watching from the Netherlands, it's really important to know that SWAB guidelines don't mention this bug at all. So you're basically left to guess, probably by looking at what you do for an enterococcus infection. What about kids? Well, the data there is even thinner, which is pretty scary. But there is this one really interesting case of a teenager with leukemia who had a bacteremia, and it just went away on its own without any antibiotics. It kind of suggests that just maybe in a stable patient, a watch and wait approach could be an option. But again, no official guidelines. So how do we stop this thing from spreading around the hospital? Easy. We treat it like VRE. The logic is simple. It's an intrinsically vancomycin-resistant bug that lives in the gut. So we borrow the entire VRE playbook. That means standard and contact precautions, gowns and gloves. It means a single room if you can. It means being absolutely obsessive about cleaning. And maybe most importantly, it means solid antimicrobial stewardship to stop giving these bugs a selective advantage. And this brings me to a really important point, and it's a call to action for all of you watching. Look, clinical experience with Pediococcus acidilactici is super limited. So we're genuinely asking you, please share what your hospital does. What are your IPC policies? Have you seen a case? Drop it in the comments. Because this is so rare, the only way we're going to get better at handling it is by pooling our knowledge. So if you got something out of this deep dive, do us a favor and hit that subscribe button. We've got a lot more explainers like this one coming up, and seriously, leave your feedback in the comments. We read every single one, and your real-world experiences with these organisms are just invaluable to the whole community. All right, I want to leave you with one final question to chew on. Probiotics are everywhere now, right? Their use is just exploding. So are we accidentally inviting this friendly foe into our hospitals and into our most vulnerable patients? Are we creating the perfect storm for this bug to cross the line from friend to foe? It's something we really need to think about. Thanks for watching.